I welcome you to this module which is related to understanding of inoculum buildup. What is inoculum buildup? When the fermentation is at the laboratory level or at the pilot plant level, the amount of fermentation media is very less, and therefore the quantity of microorganisms required for inoculation is small. But when from pilot level it is switched over to the industrial level, the microbial cells or the inoculum required to inoculate such enormous media is very high. Generally, the quantity of media ranges from 10,000 gallons to 30,000 gallons, and therefore the quantity of the organism required becomes too large. If the same amount of inoculum is used for all the levels of fermentation like laboratory scale, pilot plant and industrial level, we can easily imagine that the inoculum would be insufficient for the pilot plant and at the industrial level. Only loopful of inoculum is not satisfactory for industrial fermenters. Therefore, a requirement is generated for the scaling up of fermentation to increase the quantity of inoculum, which is called as inoculum buildup. The method of inoculum buildup. The inoculum buildup starts by using the stock culture, which is transferred to a sterile inoculum medium. Stepwise, the inoculum medium is increased in volume, and this cascade of increased volume slowly increases the microorganisms growing in it and generates the large required quantity of biomass. The initial step of inoculation from the stock culture transfers very less amount of inoculum into small quantity of the media. Generally, the transfer lists from 0.5% to 5% of the medium by volume from the previous step in the sequence. The initial phase of the growth in the liquid media depends upon which type of organism is to be grown for inoculum buildup for a particular fermentation process. If cells are bacterial cells or spores of fungi, it is first suspended in sterile tap water for transfer in the liquid inoculum medium. The bacterial spores often required shocking treatment, like in case of Clostridium, to induce germination of spores. Spores of fungi and actinomycetes are heat labile and they cannot be exposed to treatments like heat treatment. In contrast, they require fixed temperatures for germination, and they also require special medium which may induce germination faster. Germinated spores are then transferred to the normal inoculum medium. The preparation of the spore suspension of fungi and actinomycetes is done by adding suitable diluents to the surface of the agar slant culture. The diluent is to be added to the sporulated agar growth and followed by scraping the loose spores on the surface with the help of sterile lube. Sometimes addition of a wetting agent is also required along with the diluent so that the spores are uniformly distributed in the dilution media. In contrast, some stationary phase fermentations, for example citric acid fermentations by Aspergillus niger, requires the spores to be floated on the surface of the liquid medium. This is achieved by adding the spore suspension to flow down the wall of the vessel, which causes the spores to float on the surface of the liquid media. In case of non-sporulating or slowly sporulating or actinomycetes, hyphae are used to transfer the same into the liquid inoculum medium. The hyphae are obtained in fragments by scraping the agar surface with a sterile lube. Characteristics of the inoculum media. The choice of the inoculum media and the incubation conditions are very much important in building up the inoculum. The inoculum media should be selected such that it would allow rapid growth of the biomass and an increase in quantity of the microorganism. The inoculum media is not necessarily same as the production media. In process of inoculum buildup, the quantity of the product formed is not at all important. Only the biomass generation is given importance too. In case of inoculum media, the conditions are chosen such that the sporulation or cell division is favored without the accumulation of product. Special sporulating medium are used for preparation of inoculum in case of penicillin fermentation. The sporulating medium has an ability to favor rapid growth of the spore formation and the growth of the cell. Let us now see the composition of the media, which can be selected as an inoculum medium as it differs from the production media. Special considerations for enzyme production. In case of fermentation, which are targeted for enzyme production, then it is of utmost importance to incorporate the inducing substrate in the medium which is used for inoculum buildup. By this step, time is saved, that is, the lag period which is required for induction of the enzyme production by the microorganism can be avoided. Direct production of the product is achieved in the final industrial fermentation tank. Separation of cells. It is desirable to first separate the cells or dispose the inoculum media and then use for inoculation to the next step. This can be achieved by following method. Inoculum media generally should contain very less amount of carbon source. Use of less carbon source causes the deficiency of carbon as the microbial cells grow. Deficiency of carbon causes formation of floccules which settle at the base. These floccules can be drained out from the fermenter, or the above supernatant can be separated out in order to separate the cells from the inoculum medium. Or simply, methods like centrifugation, sedimentation can be used for separation of cells from the inoculum medium. The use of centrifugation or sedimentation has a disadvantage, that any particulate material present in the inoculum medium will accompany along with the cell sediment. The inoculum medium should contain all soluble components and there should be no particulate matter which would interfere with the process of sedimentation. The adoption of this method to introduce only cells has a very good effect upon the industrial production of the product. Generally, 20% of cells are used as inoculums of the final fermentation media volume. Final fermentation media is also not diluted due to addition of inoculum along with the cells. There are no alterations in the pH. Undesirable matter from the inoculum medium cannot accompany inside the industrial fermenter. High amounts of inoculum are used in bisulfite ester glycerol fermentation. There is every chance that a microbial strain, when preserved, undergo mutation during the inoculum preparation. Awareness regarding mutations. Natural strains, when exposed to inoculum buildup, there is a fast growth in order to generate high quantity of biomass during this process. There is every possibility that the mutation can take place. If the organism is a natural strain and not a mutant strain, then such problems are not so important. But the mutation during inoculum buildup takes a serious turn if the mutated strain is used for fermentation. The high frequency of back mutations have every chance during the inoculum buildup of engineered organisms. So the final inoculum can contain a larger number of back mutants which pose a big problem in the field of fermentation and will badly affect the product formation. Contamination checks are very important. Contaminations must be checked at each stage of inoculum buildup, which is very vital step to prevent the hazard in the final industrial fermentation process. The bacterial contaminations oftenly encountered when the fermentation is using hyphae of fungi or actinomycetes. Bacterial contaminations can be identified by presence of turbidity or foaming in the medium. Microscopic examinations should be used as an aid to see the broth. Any contaminating organisms in the inoculum media should be tested intermittently. Inoculum medium for anaerobic fermentation should be tested for the contamination by facultative anaerobes. It is easy to analyze by streaking the plates and growing it on both aerobic and anaerobic incubation conditions. Fermentations catalyzed by Clostridium acidobutylicum for production of acetone butanol is one such example. The characteristic of fermentation gives a hint whether contamination is present or not. Inoculum buildup, therefore, is a vital step in the production of the product in the industrial level. Following are the flow charts which will help you to understand the main steps in inoculum buildup. I hope this module has developed a good understanding about the topic. Thank you.